Hi, my name is Gilad Evrony. I'm a faculty member at the Center for Human Genetics and Genomics at NYU Langone Health and the NYU Grossman School of Medicine, and I'm very happy to speak with you today about a new technology that my lab developed to sequence single-strand mismatch and damage patterns by long-read sequencing. The background for this technology development is that all healthy cells in the human body accumulate somatic mutations throughout life. This finding was definitively confirmed with new sequencing technologies that have shown that in any tissue in the body, as we age, mutations accumulate in the genome, and that this happens in a relatively linear fashion. And you can see here one study from 2016 that shows this for cerebral cortex, kidney, and colonic epithelium. Subsequent studies have shown this with single-cell sequencing methods in neurons of the brain in the cerebral cortex, in the esophagus, in the endometrium, even in sperm and eggs. Other studies have found this in skin, liver, and lung. Every tissue that's been examined shows this phenomenon of an accumulation over time in a linear fashion of somatic mutations with age. If we actually look at these mutations more in depth, other than just quantifying their total burden, we can actually see interesting patterns that give us information about the processes that are giving rise to these aging-related somatic mutations. Specifically, we see that a sizable fraction of these somatic mutations are C to T changes occurring at CG dinucleotides that very likely arose due to a process of deamination of 5-methylcytosine. Another significant fraction, actually a larger fraction of these somatic mutations, actually doesn't have a clear pattern, and they're categorized in a mutational signature called signature 5 or SBS5, and the origin of this mutational pattern is not known. So we know somewhat about the processes that cause these somatic mutations that accumulate with age, specifically deamination of 5-methylcytosine, but most of them we actually do not know the mechanism that's causing this progressive damage to our genomes with time. You can see here a specific breakdown of these two mutational processes or signatures, signature 1 and 5, specifically in the colonic epithelium, and you can see that both of these mutational processes increase with time in a linear fashion. We can actually also see these aging-related somatic mutation signatures when we sequence tumors, because when a tumor first starts, it originates from a cell in the body that already has aging-related somatic mutations, and then when the tumor cell expands and we then sequence that tumor, we can see all of those aging-related somatic mutations that were originally in that cell from which the tumor first began. And so therefore, we can actually quantify those signatures 1 and 5 separately in different cancers, and different types of cancer show this relationship with age more clearly relative to the age of diagnosis. So for example, a stomach cancer that was diagnosed later in life has more of these aging-related somatic mutations than a cancer diagnosed earlier in life. And this is very likely because not necessarily that these mutations occurred as the cancer tumor developed, but rather these are mutations that occurred in the cell that preceded the origination of that cancer. These were aging-related somatic mutations that were occurring throughout life of this individual, leading up to when the tumor first started proliferating aggressively. Some major open questions in the field are what are actually the origins of these mutations? As I mentioned, there's a large fraction of these aging-related somatic mutations that are still unclear why they're happening. And because lightning doesn't hit both strands of the DNA twice, mutations don't spontaneously arise usually in both strands of the DNA. They actually first begin as a change to one of the strands of the DNA before it becomes a double-strand mutation. We'd really like to know what are the single-strand origins of mutations. We'd also ideally like to study this process across many variables, different organs, more than just those that have been examined so far, different cell types may have different aging-related somatic mutation rates and patterns. We'd like to understand the influence of the environment and exposures on somatic mutation rates as we age. We'd like to know whether these may vary across individuals in the population. Are they predictive of cancer risk? And perhaps the most provocative question that's been debated for a long time is, what is the relationship between these aging-related somatic mutations and aging? Is this just a biomarker, a correlate of aging, or do these somatic mutations actually cause physiologic aging to some degree? And answering these questions requires an ability to measure somatic mutation rates accurately and scalably, which is actually not possible to do with standard DNA sequencing. And the reason for this is that standard short read or Illumina sequencing has an error rate of about 1 in 1,000 bases. But these somatic mutations related to age that I've been talking about are present in any given sample at a level of about 1 in 10 million to 1 in 100 million bases. So the signal level of somatic mutations related to age is way below the noise level of standard sequencing error rates. And this is why specialized methods have been required to actually study the process and discover those things that I showed you in the prior slides. Specifically, single-cell sequencing has been a major approach to be able to detect these somatic mutation rates, because when you get down to the level of a single cell, you can now see that cell's individual somatic mutations, even though they're at very low levels on a tissue level. Duplex sequencing is another approach that has allowed standard DNA sequencing to radically increase its accuracy. And the way this works is through attaching unique molecular barcodes to double-stranded DNA molecules right at the beginning of the process, where those barcodes are actually reverse complements of each other. And then following sequencing of copies of each of those strands, single strands of DNA, we can computationally reconstruct, using these barcodes, which strands of DNA originally belonged together in the original double-stranded DNA molecule. And that allows filtering of all of the amplification artifacts and sequencing errors, leaving true double-stranded DNA mutations. Another approach is to sequence a population of clonally related cells that can be microdissected out of tissue. This is basically nature's way of amplifying DNA for us accurately, so that we don't have to do single-cell sequencing, and we also don't need to do this highly accurate duplex sequencing to avoid amplification artifacts. However, all of these methods do not actually interrogate the original DNA molecules. They all involve some form of amplification, including this microdissection method, which still requires PCR amplification of the DNA. And because the original DNA molecules are not directly interrogated, we actually cannot see true single-strand changes to the DNA, because the amplification process introduces too many single-strand artifacts. And so we set out to develop a method that performs direct single-molecule sequencing of DNA while also getting to this ultra-high accuracy that allows us to see these very low-level somatic mutations related to age. We reasoned that we could take long reads, long pieces of DNA that are long relative to standard short reads, but actually are shorter relative to standard long read sequencing that usually sequences DNA that's about 20,000 base pairs in length. Instead, we process DNA that's 1 to 4,000 bases in length. We attach hairpin adapters that topologically circularizes the DNA. This is a standard approach of Pacific Biosciences or PacBio single molecule sequencing. The only difference being is that the DNA is shorter than the standard PacBio sequencing read length, but it's still longer than standard short read DNA sequencing. And then the PacBio sequencer is seeded by these molecules, and the polymerase of the sequencer goes around and around this molecule many, many times, and we can then filter out sequencing errors that happen in each of these passes of the molecule, leaving us with a very high accuracy to detect true double-strand mutations that are found in every pass of the molecule relative to single-strand true events that are found in only passes of one of the strands. When the polymerase goes around and around this molecule, it first sequences the top strand, then the bottom strand, then the top strand, then the bottom strand over and over again. So we can clearly distinguish double-strand events from single-strand events. And this technology was developed by two very talented members of my laboratory, Meihong Liu and Benjamin Costa. And again, in this situation, we're directly sequencing the original DNA molecules from an individual, from a tissue, without any amplification. This is what the data actually looks like. One single molecule's data looks like this in the top half of this diagram. And you can see in blue are the individual passes of the top strand, and in red are the individual passes of the sequencer for the bottom strand. And you can see each different color is a difference from the human genome reference, and you can see random colors sprinkled throughout. These are sequencing errors, while you can also see these vertical streaks of color that were changes to the DNA relative to the human genome reference that were seen over and over in nearly all or all of the passes of the molecule in both strands. In the bottom is standard DNA sequencing, short read sequencing of the same sample. And so you can see that most of those vertical streaks are true double-strand mutations that were also detected in the standard germline sequencing of the individual. But there is one area here highlighted that I'm going to zoom in, where this double-strand DNA change was not seen in the standard sequencing of the individual. And this is a somatic mutation because it was not detected by standard germline sequencing of the individual. So we wanted to now confirm what is our actual accuracy. Did we get to this level of at least 1 in 100 million errors per base, which would then allow us to detect these aging-related somatic mutations? And to do that, we used a tissue sample that has the lowest mutation burden of any tissue in the body, which is sperm, actually except for eggs, which have a lower mutation burden, but eggs are not readily accessible. And so we can profile sperm with our method, which we call high-def-seq, which stands for hairpin duplex enhanced fidelity sequencing, and we can compare it to the next gold standard method in the field called nanoseq, which is a short-read duplex sequencing method. And we can also compare it to estimates of sperm mutation burdens based on de novo mutation studies, where you count the number of mutations a child has that the parent did not have, and you specifically look at those mutations that are phased to the father. It gives an estimate for the mutations that were brought into the child from the sperm. And you can see that our method, the circles, have indistinguishable mutation burden relative to nanoseq and relative to these de novo mutation studies, and that we're successfully detecting a somatic, in this case, a mosaic mutation burden of less than 2 mutations per 100 million bases. So indeed, we were able, with single molecule sequencing for the first time, to reach ultra high fidelity using this PacBio sequencing technology. To further confirm the accuracy of our method, we profiled samples that have a predisposition from individuals who have a predisposition to cancer. They have a mutation in their body, in every cell of their body, that causes an increased level, increased rate of somatic mutations, and these mutations happen with a particular pattern. So this individual that we profiled has a mutation in the polymerase epsilon protein that's responsible for actually copying the genome during DNA replication. And you can see at the top, our method recovers a pattern of mutations that is indistinguishable from nanoseq double-strand DNA mutations. And you can specifically see this breaks down the mutation pattern into 96 possible mutations based on which base is changing to which other base. So this further confirms the accuracy of our method. And again, up till this point, actually, this data is only looking at double-strand mutations, which prior technologies have been able to do. We then looked at a variety of tissues and saw this linear increase with age in double-strand DNA mutations. This is expected, and I showed you that data before from other studies. It's also interesting to see in our data that liver has a higher mutation rate than other tissues, especially relative to the brain or neurons from the brain. We're also able to see a difference, we're also able to see the different mutation patterns, the two signatures, signature 1 and 5 that I mentioned before, with one outlier sample of an individual with specific kidney immunosuppression due to a transplant. You can see here now is where we see new types of biology that have not been seen before by other ultra-high fidelity sequencing methods, specifically single-strand events in the DNA, changes to the DNA that are only present in one of the two strands. And you can see that clearly in the data here, a change in only the top strand of this DNA molecule. It was also not seen in both the bottom strand and in standard DNA sequencing of the individual. To further confirm our ability to detect single-strand changes to the DNA, we profiled more samples from more cancer predisposition syndromes, and we detected an increase in single-strand DNA changes in individuals with mismatch repair deficiency and with these polymerase proofreading mutations like I showed you before. You can see that in purple and in red here has an increased single-strand DNA call burden relative to healthy tissues. We further can now look at the pattern of single-strand calls, and at the top you can see the single-strand calls of an individual with a polymerase epsilon mutation relative to the double-strand DNA mutation pattern of that same sample. And at the top, because we're now looking at single-strand calls, we actually have to show the pattern of all possible 192 changes to the DNA. We color the two-strand changes in purple and in orange. And you can see that this double-strand DNA change, this peak, for example, at the left of TCT to TAT, at the double-strand mutation level, where we actually don't know if it's actually a TCT to a TAT or if it's an AGA to an ATA, at the top, the single-strand pattern actually tells us it was an AGA to an ATA. It basically resolves for us which strand this double-strand mutation is arising from. That gives us information about the origin of this double-strand mutation pattern. This actually matches, the single-strand pattern matches what has been seen before in yeast and in in vitro polymerase studies of polymerase epsilon. We further profiled tumors from individuals with germline inherited mutations in mismatch repair proteins, along with somatic mutations in polymerase epsilon. These tumors have an ultra hypermutating phenomenon where not only are they getting a lot of mutations due to errors of the polymerase, but they're also deficient in mismatch repair that fixes those errors. So these tumors have an incredibly high mutation burden. And you can see here two tumors we profiled, a medulloblastoma and a glioblastoma from these individuals. And you can see an incredibly high mutation burden on the left at the double-strand level. And then we also see a very high single-strand call burden and correspondingly higher single-strand burden for the tumor that has a higher double-strand mutation burden. And we can now also look at the patterns at the single-strand level and the double-strand level of these tumors. And for one of those tumors, the glioblastoma, you can see here additional peaks that were not seen before in the presence of just a polymerase epsilon mutation, because now we're also seeing those mutations that are arising due to the deficiency in mismatch repair. At the top is the single-strand calls, and the bottom is the double-strand mutation calls of this glioblastoma tumor. And you can see, again, a correspondence in the peaks between the single-strand calls and the double-strand calls, but the single-strand calls are actually resolving the strand from which the double-strand mutations are arising. Looking at single-strand calls across a panel of healthy tissues, we can actually see another interesting phenomenon, which is an outlier level of C to T single-strand calls in sperm. Not only do they have a higher C to T call burden, but also a larger percentage of all of their single-strand calls are C to T. You can see here the pattern at the top of these single-strand calls in sperm, which actually matches the pattern that we saw when we simply damaged DNA by heating it. So heating up DNA has always been known to cause cytosine deamination damage, and now for the first time we can actually see the pattern of that damage directly and find that it matches an endogenous form of C to T calls in sperm, suggesting that these sperms actually harbor cytosine deamination at a high level. This may be because sperm do not have a highly active DNA repair process because their DNA is highly compacted, so these cytosine deamination events may be accumulating over time without the capacity to repair them. And then presumably, when the sperm fertilizes the egg then repairs that damage. Furthermore, PacBio single molecule sequencing is really fascinating because it not only provides us information about which nucleotide is incorporated, but also the kinetics of that nucleotide incorporation, the time it takes the sequencing polymerase to incorporate the DNA, which is called the pulse width, or PW in the diagram on the top left, and the time between nucleotide incorporation events, which is the interpulse duration, or IPD. Interestingly enough, we see a difference in the kinetics of these C to T single-strand calls in sperm samples relative to C to T double-strand mutations in regular tissues. This makes sense because these C to T calls at the single-strand level of sperm are not actual changes of cytosine to thymine, but rather they are damage to cytosine that's causing them to sequence as if they were thymines, but actually they're deaminated cytosines, likely uracils. We now know from other experiments that indeed these are uracils. It's true cytosine deamination into uracils. And that's why there's this altered kinetic profile. I'm showing here this change in color-coded of pulse width duration in sperm samples, specifically in the two bases following the actual cytosine deamination base. Another interesting finding from our technology is that we were able to, first of all, see the expected increase in the mitochondrial genome somatic mutation burden. It's been known for a long time mitochondria have about a 40-fold higher double-strand mutation burden relative to the nuclear genome. Our technology sees that and confirms that in the liver and kidney, which have a large number of mitochondria per cell, which is why we're able to profile this. However, surprisingly, at the single-strand call level, we do not see a difference between the nuclear and mitochondrial genome. This is in fact surprising but consistent with recent studies that have suggested that the process of mitochondrial genome mutation actually occurs during replication, in which when the DNA of the mitochondrial genome is denatured into single strands, it incurs damage at that point that is then rapidly fixed into double-strand mutations, so that the time between the single-strand damage and the double-strand mutation fixation process is short, and that's why we actually don't see those events. In addition to the fact that our technology only captures double-strand DNA molecules, we don't capture single-strand of DNA, so our data is actually consistent with the mutation process of mitochondria happening during mitochondrial genome replication. So to summarize, every human tissue accumulates mutations with age, and measuring these requires ultra-high fidelity sequencing. We've developed the highest fidelity DNA sequencing method to date for single base changes, and we resolve for the first time not just double-strand changes to DNA, but also single-strand changes in single molecules. The single-strand mismatch patterns we see reveal the origins of mutations in some cancer predisposition syndromes and ultra-mutated tumors, and mitochondria have a surprisingly low single-strand mismatch burden. Looking forward to next steps of what we would like to use this technology for, we'd like to study mutation rates and patterns in different cell types. We'd like to study indels. Currently, this has been very challenging because the error profile of single molecule sequencing for indels is too high for us to resolve true indel changes, but we're working on computational methods to try and address this, but we may not be successful. And in the long term, we'd like to study somatic mutation processes in larger numbers of people and phenotypes in the context of a variety of diseases. So there are many people to thank who are participants in this project. I especially want to mention Mei Hong Liu and Benjamin Costa, as well as other members of my lab, Marta Gronska-Pesky, Rachel Bandler, and Yuna Choi. Other collaborators, Jonathan Shoag from Case Western, Uri Tabori from SickKids Hospital, and the Cryo Sperm and Egg Bank, as well as the NIH Neurobiobank, as well as collaborators and mentors at the NYU Grossman School of Medicine, Aravinda Chakravarti, Ben Neal, and Jeff Buka. You can visit our website for more information about what our lab does, and I hope you enjoyed this talk, and happy to be in touch if you have questions. Thank you.